Thank you for the introduction, and thanks to Oxford Nanopore Technologies to give me the opportunity to present our research on direct RNA sequencing in clinical diagnostics. So, first of all, we have heard already why RNA modifications are so interesting. So they play multiple crucial functions in multiple biological processes, especially transcriptional regulation, including RNA stability maintaining and translation or RNA splicing. And it has been found that this regulation, as we have already heard, is present or has been found in multiple diseases like neurological diseases, as well as immunological diseases, or metabolic diseases, as well as cancer progression. And this is why RNA modifications are getting more and more attention as clinical targets in RNA therapies, as well as for modopathy diagnostics. And today I will present the first clinical example of how we could use direct RNA sequencing to solve a patient case. And in our Institute of Human Genetics, we had one patient of 1 year old, and it has intellectual developmental delay and severe microcephaly. So first we did our routine clinical approach of sequencing the blood sample by whole exome sequencing. And what we have found were 2 compound heterozygous variants in the METL5 gene. One variant was already known as pathogenic, and the second variant was a potential splice variant and classified as of unknown significance. The METL5 gene is a methyltransferase, an RNA-modifying enzyme that adds specifically one methylation at the 18S rRNA at the position 1832 and leading to an M6A. And this position is very close to the decoding center of the ribosome and is thought to influence global translation as well as transcript-specific translation. And we already heard why modifications in our RNA are very important and influence many processes. And yeah, loss of function or loss of methylation of this specific site is associated already to severe microcephaly as well as global developmental delay. But however, we have one already known pathogenic variant of this autosomal recessive disease and one variant of unknown significance. So we cannot be sure already that these 2 variant combinations are the cause of the disease. So how can we confirm the genetic cause of these variants? And here direct RNA sequencing comes in, because we can use direct RNA sequencing to detect both the splice effect as well as the RNA modification loss at the specific site because of the feature of sequencing the native RNA. And here's a short timeline of flow cells and chemistries developed by Oxford Nanopore Technologies. So the most popular flow cells that were developed are the 002 and the 004 flow cell, whereas the 004 flow cell is higher in throughput, accuracy, and quality. And together with the latest flow cell, Oxford Nanopore Technologies also developed de novo RNA modification models, including M6A. And for the older flow cell, which is discontinued, third-party tools are only available, including the M6A basecaller, which we already heard in the first presentation, as well as MAFIA and other basecalling models. So with the goal in mind to solve our patient case, we first want to validate the performance of the recently released new 004 flow cell in comparison to the 002 flow cell. Next, we wanted to investigate how good is the performance of M6A detection by the production basecaller Dorado, and in comparison to the 002 based methods, and gold standard ChloriSeq technique. And last, we show the potential of the usage of direct RNA sequencing in our clinical patient. These were the samples we were using. We had 2 human cell lines as well as human blood samples, and also samples that were in vitro transcribed, so with removed RNA modifications as an unmodified control. These were sequenced both with the 002 and the 004 flow cells, and this was our bioinformatics pipeline. So coming to the general performance results, first of all, we could detect that 004 samples in all different tissue types was higher in throughput compared to the 002 data. This trend was also observed for raw read accuracy as well as person reference identity. And looking at the different base calling errors, we also observed again that the frequency of mismatch, deletion, and insertion was much decreased in the 004 data compared to the 002 data. We also did a comparison with all available, or the available base callers for 002 and the 004 Dorado base caller, and still Dorado outperformed all the other base callers that are also coupled with the RNA modification detection models. Now coming to the RNA modification analysis and the performance of 002-based and 004-based models. For 002, we used the community-driven models Mafia and M6A base caller, and for 004, we used the Dorado RNA modification detection model. And comparing the sites between 002 models, we observed a relatively low overlap between Mafia and M6A base caller. However, Dorado could detect all of the M6A sites from either 002 detection tool and many more sites. We also sequence our blood samples by ChloriSeq, which is a gold standard RNA modification technique based on an enzyme or chemical method. And we observed for direct RNA sequencing and GLORY a couple of uniquely identified sites, however, a relatively large overlap of direct RNA sequencing in GLORY, and 85% of the GLORY sites were also detected in by direct RNA sequencing using the 004 flow cell. And we also showed a relatively low number of false positives, as shown by the detected sites in the unmodified IVT. When looking at the methylation frequency between GLORY and direct RNA sequencing, we saw a slight decreased median M6A frequency, so the stoichiometry in the direct RNA sequence data were slightly decreased. However, when looking at the correlation between joint sites of GLORY and RNA 004, we observed a correlation of 0.92, and also the correlation between replicates of GLORY and RNA of two RNA 004 samples reveals a high consistency of 0.95 correlation coefficient. And with this, we conclude that Dorado was able to detect a high number of sites detected also by the gold standard method, and is relatively accurate and reliable. And this is why we went for direct RNA sequencing in our patient case. And first of all, we could validate the splice effect shown by an exon skipping in half of the transcripts, and also the transcript that had the exon skipping also didn't have the pathogenic variant. And lastly, we could show that we have a loss of function in the M6A site of the 18S rRNA, which is shown in the bar plot on the right side, where we had a lower than 1% M6A frequency in the IVT and the patient sample, and a frequency of over 14% in our blood control samples. And yeah, with this, we could conclude that based on our direct RNA sequencing results, the genetic mutations are likely the cause of the disease of our patient. In summary, 004 definitely outperformed 002 in throughput, read quality, and accuracy. We also saw a high concordance of M6A sites derived by Dorado in comparison to Glory sites as well as replicates. And but due to a relatively high number of potentially false positives as well as sites that we haven't detected but were detected in Glory, further benchmarking is needed. And but the site-specific M6A calling was able to validate the loss of function in of genetic variants in our patient case. And we believe that especially site-specific RNA modification has the potential to increase the diagnostic yield and assist to clear unresolved patients in diagnostics. And yeah, with this, I would like to thank all the people that were involved in this project, especially Charlotte Hevel, Johannes Miedema, and Susanne Gerber, and all the collaborators, of course. And yeah, of course, Oxford Nanopore Technologies are research use only. And thank you for your attention. I'm happy to answer questions.